by one. My name is Farida. What is your name?
Harshini.
Okay, Harshini. Hello, how are you doing?
Fine, auntie. How are you doing?
I am also doing very good. Nice. Okay. Who else is there?
Me. My name is Samir.
Hi, Samit. How are you?
I'm good. I'm doing well. Thank you.
Good. Nice. So we have Harshini, we have Samit. Who else?
Ma'am, me Atharv.
Yes. Atharva, if you want to see your face, come on, show me your face. You can switch on your camera.
Yeah.
Hi, Atharva.
So
So Atharva is a very old student. He's been with us for almost 3 years, 4 years. Harshini, Samit, Atharva. See, it will be so nice, if because we all know each others, we'll be there throughout the year together. So if you know your co-students, it is going to be very nice. Who else is still missing? There's some here. Who is here?
Hello, ma'am.
Okay.
I'm here.
Yeah, hi here. How are you? Can we
Hi,
see you?
ma'am. I'm fine.
Yes, yes, good, good, very nice. Okay, so we'll begin with our class today. Okay, see now, as ma'am explained, I will explain you how the procedure we'll be taking the class. Okay, first I'll be introducing you to the topic. I'll be explaining the topic. So we'll be taking around 10 to 15 minutes on the topic, and after that there is another ma'am who is going to perform the experiment. So she'll be performing the experiment. I'll tell you when you have to pin yourself to her cameras. So when you'll pin yourself to her cameras, what will happen is you'll be able to see the entire process of how you have to see the experiment. Then we'll tell you to perform the experiment. Once you have performed the experiment, we'll see the observation done by you, and then we'll have a small quiz round for you. Okay? Okay, and so this is how we'll be conducting our classes. Okay. Am I audible to everyone? Yes?
Yes,
Okay.
ma'am.
So good.
Yes,
So
ma'am.
now there is okay. So now there's a presentation that is going on. You have to pin yourself to KitKat Lab. So when you'll pin yourself to KitKat Lab presentation, you'll be able to see the presentation. Can you see the presentation?
Yes, ma'am.
yes,
Yes.
ma'am.
Harshini and Atharva and Samit. Samit, yes.
Yes,
All four of you. Okay? Okay. So today we're going to talk about nutrition and plants. See, nutrition is a very important topic because we need to understand the way the plants are uh, getting their food. Okay? And plants have a very unique system of getting their food. So what do? You all must be knowing the process name by now. Do you all know the process's name? Anyone? Tell me.
Photos.
Photosynthesis.
Very good. Photosynthesis. Now, photosynthesis, as the word sounds, and as we have learnt when we were small kids, is not as simple as it is. It is very much complicated. We'll be studying a little bit one level up above than what we have studied before. Probably when we were kids, we were told photosynthesis was when the green plants, when there's sunlight and they take carbon dioxide from the atmosphere, they produce the food that is in the form of glucose, and they produce also oxygen from it. Correct? So it is a little bit more deeper to that. We'll be studying about it. Okay? So we'll go to our first slide. So we'll go to the next slide. Okay. So see, food is very important. Can you all live without food? No. Even in lockdown also today we are very hungry and we need to eat more than probably when there was no lockdown, right? Your mother is making more variety at home. So food is a requirement, very important. We need to consume food. Okay, the same way all living things, even plants are living things, right? So all living things will require food. Okay, and nutrition is very important. Why do we require nutrition? For many reasons. First is to grow. When you were a small kid, you were given milk. Then when as you grow older, you were given mashed food. Then slowly you started eating the food which you started chewing as per the stages. That same way, food is very important to grow. You're going from small to big. The same way, a plant also is growing from a small seed to a big plant. So it requires food at different stages. Okay. Secondly, to repair. Now, if supposing there is some part of your body that has got injured, you fell off the cycle. So what will happen is there is a wound out there. Now that wound needs to be repaired. So that repairing is done by the food, by the energy that is provided by the food. So you need to have good and healthy food. Okay. Plants are unique. Plants can make their own food. Plants don't have to depend on somebody that they will cook the food for them and then they will be eating it. No. Plants make their own food. Okay. Animals, any animal, what do? They either take their food from plants. If they take the food from plants, then we call them as herbivores. If the plant is taking the food from another sorry, if the animal is taking the food from another animal, then we call that an animal as a carnivorous, right? So that way animals also get their food from either plants or from other animals, right? And human beings, we also either depend on plant or we depend on animals for our food. But indirectly, you all must have studied the food web. Have you all studied the food web? What is a food chain? Do
Yes.
you know what is a food chain? Yes. What is a food chain? That the first producer is the plants. The plants are the ones that are producing the food from the sun. So the sun is providing the energy because of which the plants are taking the food. From the plants, the animals take the food. Those are called as the secondary consumers. First is the primary producer, the plants. Then we have the secondary, that is the animals which take from plants. And then the tertiary ones would be the ones, the animals that take from the other animals. So we come in that category, right? So directly or indirectly, the human beings are dependent on plant itself. Okay. So on based on this we say that nutrients are important. So what nutrients are there? So there are for human beings, if we consider, there are a few categories of nutrients that we have described. Okay. For example, we have described what are carbohydrates, what are proteins, what are fats, vitamins, minerals, water, and roughage. Okay. These 6 to 7 main macronutrients are the ones that are important for us. Okay. So carbohydrates we get from, say, bread, or from starchy food, from wheat. All this gives us carbohydrates. Carbohydrates are very important for the major growth. For the major growth, carbohydrates are important. Then next will come your proteins. What are your proteins? Proteins are very important because each and every cell in your body requires those proteins for making a lot of other chemicals. For example, enzymes, or some kind of RNA, DNA processes, some other proteins which are required for some other part of the body. So proteins are very important. Fats, fats are important so that there is an anti-greasing mechanism, like there's a greasing mechanism in your body. A little amount of fat is important in your body. Not more or not less, but the exact quantity of fat is important. Then we have a lot of vitamins and minerals. Now, what are these vitamins and minerals? Vitamins would be like vitamin A, vitamin B, vitamin C. These help in the processes of making a lot of items which we are using in our day to day life. So, that is very important, vitamins. And minerals like calcium, potassium, and so on. Now, what is the work of water and roughage? Water and roughage is mainly used for creating or for helping your body to remove the unwanted wastage from your body easily. If, for example, you don't have roughage, there is a lot of constipation problem, because of which it can lead to many other diseases. So each and every quantity in a proper requirement is needed by a body. So there is something which is called as a food pyramid that we follow. I'm very sure you must have done it before, food pyramid. Okay, so this is a kind of a revision of what you all have done up till now. Okay, next slide. Okay, so nutrition, if I want to define what is nutrition, nutrition is the mode by which the food is taken by an organism and it is utilized by the body. So it is not just eating it, not just eating, it's a very long process. The after eating it, after it goes into your body, into your digestive system, how your body is digesting it, how your body is utilizing it, that entire process is called as nutrition. And the nutrients, as I discussed, are carbohydrates, proteins, fats, vitamins, minerals, roughage, water, and so on. Okay. Now, based on nutrition, there are two types of organisms which we broadly classify. Okay. One is autotrophs. What are autotrophs? The word auto itself means on its own. Okay. So when I say auto, it means that they can produce the food by their own mechanism, by their own. Example, plants. Only plants. There is no animal which can produce food by their own way. So it is only plants, autotrophs. Okay. Now we have got heterotrophs. What are hetero? Hetero means different. Hetero. So they are depending on different organisms for their food. Such organisms are called as heterotrophs. So on broad classification, we've got autotrophs and we have got heterotrophs. Okay. Autotroph example will be plants. Heterotroph examples will be animals, and also some plants which we call them as carnivorous plants also. Okay. Next. Okay. So today we're going to learn everything based on photosynthesis. So we're going to study the process of photosynthesis more deeply today. Okay. Basically, if I want to tell what is photosynthesis, it is a process by which the green plants are making their food. And what they require? They require carbon dioxide. So carbon dioxide is available where? In the atmosphere. So they're going to take carbon dioxide from the atmosphere and they're going to take water. Water will come from where? Water is there in the atmosphere, but not the quantity that the plants require. So the major quantity of plants that they require for photosynthesis process will come from the soil. So from the soil, from the roots are absorbing the water, okay, and the parts of the plant, they are going to absorb carbon dioxide. These two things are going to combine and they're going to make food for the plants, which comes in the form of glucose. And in that process, they're releasing one byproduct, one product which they don't require at that time is called as oxygen. Okay. So oxygen is released and that is given out in the atmosphere. This entire process is called as photosynthesis. Okay. Now, where this process is happening, very important. Okay. As we were kids, we were always told that this process is happening in the leaves. Okay. We call the leaves as the food factory, or we call the leaves as the kitchen of the plant. Okay. But food also happens in different parts of the plant that are green. So, mind you, any part of the plant that is green will carry out the process of photosynthesis. Now, I want you to think and tell me which other part of the plants are green, or give me some example of some part and tell me in which plant that is green. Come on, tell me.
Ma'am, STEM.
Give me example of the plant.
Uh,
Ma'am, it's
Because see, normal.
he said correct that it's stemmed, when the trees are sapling, when there are saplings, then the stem is green.
Ma'am, in
Okay, well,
shrubs or even in
yes, in
it
small
all.
shrubs also. Yes, small shrubs and herbs. Herbs, we eat a lot of herbs, no? We eat coriander, we eat mint. All those stems are green in color. Correct? So
Yeah.
stems, leaves. What else? Think. What else? You all have seen flowers. Which part of the flower is green in color? Have you all seen any part of the flower green in
Ma'am,
color?
sepal. Ma'am, sepals.
Yes, very good. The sepals. The sepals. So one of the function of the sepal is also to carry out photosynthesis. Okay. So the sepals can also carry out the process of photosynthesis. So keep this in your mind. All the green parts of the plant will carry out the process of photosynthesis. Now, why it is so and how it is so, we are going to study it further. Okay, hold on. Okay. Now this transportation of water, as I told you, is happening by the first part of the plant that is in contact with water is your roots. So the roots are taking them from all the soil, from the soil. So the roots are going very deep down. The roots go very deep down so that uh, they can pull up the water from wherever water is available. Okay. Now along with water, they don't only take water. Okay. Along with water, they will also take minerals like sodium, potassium, calcium, magnesium, and so on. These minerals, they are dissolved in the water. So they are not going and specifically picking up these things. They are automatically there in the water. The water out there in the soil contains these minerals. So they are picking up the water and these minerals are dissolved inside them. So these things are picked up by the roots and they are going up into the stem. So the stem is pushing it up. How it is pushing it up? It is done by a process called as capillary action, because the tubes which are pulling those water up are very thin, so they are able to pull up the water. And there are many other factors also involved. Okay. That we'll study when we come to that part. Okay. Right now just understand that the roots are picking up the water, the stem is taking it up, and from there it is going to any green part of the plant. Right now, for example, let's consider leaves. Okay. So they're going to the leaves. Okay. In the leaves you must have seen the veins. You must have seen the big main vein, then there are small veinlets. So that is where all the water is being transported. Okay. So this is very important. From the roots and via the roots and stems, we get water. Now, secondly, we require carbon dioxide. So carbon dioxide is there in the atmosphere. Now, for it to come from the atmosphere, what is it that the plants will have to do to get it inside it? See, pulled it up. Roots pulled it up through the tubes I told you. Now, what should be there in the plant, the green parts, that it should take in the carbon dioxide gas? It is a gas, right? Water was liquid, it was pulled up. Now, this gas has to come inside the green plants. What can it be? Can you think and tell me? Guess it. It could be wrong, it could be right. It's okay. Come on, Harshini,
Ma'am,
Priya.
nitrogen. Ma'am, nitrogen.
No, 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 no. I'm asking how carbon dioxide from the atmosphere. See, carbon dioxide is all around us. Same way, carbon dioxide is all around the plant also. Now, the plant has to take the carbon dioxide into it. How can it do that? Can you think of a way? For example, you think you are a plant.
Ma'am,
You
pores are
think
Ma'am,
you are a
there.
via stomata. Ma'am, using stomata.
Ma'am, pores,
okay.
yeah.
So y'all know what is stomata, right? Y'all know what
Yes.
is
Yes,
stomata.
ma'am.
Okay. So they are small pores. They are small tiny holes which are there in the green parts of the plant, which are there in the green leafy parts of the plant, right? So what happens is in that pores, in that small things, the carbon dioxide gas is taken in. Okay, it is taken in. Now how is it taken in? How is it taken in? There is a proper mechanism by which it is taken in. So there are small, they are called like doors. Okay, so they are doors. Okay, they are like this, they are like doors. Okay, so when they have to take in, they will open up a little bit. Okay, so there will be opening up mechanism, and when they open up, the carbon dioxide from the atmosphere will come into the leaf. Okay, once they have taken it, now they'll close it again. Okay, so there is opening and closing mechanism of these pores. The name of the pores are called as stomata. Okay, next week we'll be doing about stomata. We'll be discussing and be seeing in detail the structure of the stomata. Okay, so these stomatas, they open up and they are going to close. This opening and closing of the stomata is going to happen at different time of the day. Okay, in the day they'll be mainly open, in the night they're going to close. Okay, why? This question I'm going to ask you next week again. You all are going to come up with an answer. You all are going to search for this. Okay, the stomatas, they mainly remain open during the day, in the night they close up. Why? Okay, now these small pores, stomata, they are helping for the plants to gather the carbon dioxide from the atmosphere and take it into the green parts of the plant. Now, in the green part, now we come to the main topic. Why the green part only? Why not a red part? Why not a blue part? Why not some other part? Why not a brown part? Because the green part of the plant is because of a substance called as chlorophyll. There is a pigment, there is a liquid. That liquid is called as chlorophyll. Okay, now that chlorophyll is present in only green parts of the plant in a particular cell. If you all know what is a cell, in the plant cell there is a small portion which we call as chloroplast. There is a, let's consider a cell is like a city. Let's consider the cell is a city. One building of that, we'll take it to be as chloroplast. In that one building chloroplast, there is a liquid filled which we know, which we call it as chlorophyll. So in the chloroplast there is chlorophyll. Now this chlorophyll, why is it important? Why chlorophyll? Why not brown? Okay, my question was that. The the answer to it is because the chlorophyll is able to capture the sunlight. Now, if you have studied about chemical reaction, can I just bring two items together and just combine them? Can that happen? Tell me.
Yes.
Yes? Can I just take two things? I'm just keeping two materials. Okay, I have got right now these two things. Okay, I'm just going to mix them. Will it get mixed?
No, ma'am.
No.
Correct. I need to, what do I need to do to it? What do I need to do to it? What do I need to do to it? If I want a chemical reaction to happen, what do I need to do to it?
Ma'am, heat it.
Some kind of energy, right? So the
Yes.
one form of the energy that we can give is to
Apply
heat
energy to it.
energy. Yes, so
Ma'am, you
we need to give it.
apply energy to it.
Yes, you need to apply energy to it. So the energy that you're going to apply here to the plants is heat energy coming from the sunlight. Now, who is helping us to take that heat energy from the sunlight? Your chlorophyll, pigment chlorophyll from the chloroplast. Okay, so the chlorophyll is taking the sunlight. Okay, and the roots are taking the water, and the carbon dioxide is coming from the atmosphere. So we have got three things. We have got the atmospheric carbon dioxide, we have got the water that is coming from the roots and the stem, and you have got chlorophyll. Okay, these three things are now going to combine, and because of that they are going to now undergo a complex chemical reaction, very very complex. Okay, there's a lot of studies that is done on it. So people study it about it. There is something that is called as Krebs cycle. After Krebs cycle there's something else, and there is lot many things. Okay, for right now we'll just try to understand these three factors are important: water, carbon dioxide, and your chlorophyll. These three things are combining in that cell, and they are making the food which the plants are using. Okay, next slide. So this is how it is. The water is coming from the roots and the stem. Carbon dioxide is coming from the atmosphere. You see on the left side. Okay, and the sunlight is being picked up by your chlorophyll. These three things we are combining, and after its combination, they are making two things. After its combination, they are making two things. The two things that they are making is what we are calling them as oxygen and sugars. That sugar initially, the first time, it comes in the form of glucose. How much ever is utilized by the plant is utilized. If it is not utilized by the plant, then one glucose combines with the other. Then the third one will come, fourth one will come, fifth one will come, and so on. The glucose will start combining with each other. Once all the glucose will start combining with each other, they get converted into starch. Okay, so basically they are getting stored in the form of carbohydrates. That is what we call as sugars. Okay, the other word, chemical word for it would be sugar. It's not the sugar that you're referring to, the sugar that you eat. Okay, so we call that as sugars. So the glucose first, what in one particular reaction, see, the plant has got millions of cells. Okay, millions of cells, and out of those million cells, imagine 500,000 of the cells are green in color. So in each and every particular cell, this process of photosynthesis is happening throughout the day. I'm talking just 1 second. In just 1 second, one molecule of water, one molecule of carbon dioxide is getting converted to glucose and sugars. Okay, so imagine 500,000 cells are making how many such glucose molecules throughout the day. Whatever is utilized by the plant is utilized. Whatever is not utilized is then getting converted into starch. Sometimes they store it in different places. Like for example, in potato, they go and store it in the potato, and that is how we eat potato, and we say potato is starchy, potato is sugary. Okay, so that is what is happening in the process of photosynthesis. Is this clear?
Yes, ma'am.
Yes. Okay. One keep your this one misconception. If you had before, it's not only leaves, anything. So basically, even a cactus also will do the process of photosynthesis. Cactus, they don't have leaf, but they've got green color. The stem of the cactus is green in color, and the leaves are getting converted into spines. So that is what they process. They do the process of photosynthesis also. Okay, next. Okay, now is the experiment time. I'll tell you what you have to do in the experiment, and then I'll tell you where you have to pin yourself to other ma'am's video so that you can see the process happening. Okay, we'll first see what is the experiment that you have to do. Next page. Okay, So in this, you are trying to find out what the leaf is containing. Okay. So that is your aim. Anytime, whenever we do scientific experiments, for carrying out scientific experiments, you always go by certain scientific ways. Okay. So the first thing is you have to write the aim of the experiment. So aim of the experiment is very, very important. Okay. So you have to write the aim of the experiment. After that, you're writing the items that you're requiring. So here the aim of the experiment is to understand what the leaf is composed of, to understand what the leaf is actually containing. Okay. For that, you're requiring beaker, test tube, candle, iodine solution, alcohol solution, and test tube holder. Okay. Some other requirements that we require would be a leaf, water, and a small plate. What are you going to do? Okay, as you can see, there is a there's a tripod stand. On the tripod stand, there is a beaker containing water. This water needs to be hot. So for making the water hot, we supply candles. You can do it at home very easily. You have to just take a hot, you have to take a container containing hot water. Okay, and in another small transparent kind of a container, another small transparent kind of a container, we put alcohol solution into it. Okay, now we don't heat alcohol directly, we do this kind of double boiling method. Why do you carry out double boiling method? Can anybody explain to me why we can't heat alcohol directly on the fire? Why do you have to use this method? Anyone? Come on,
Ma'am, because
think
alcohol
logically.
is very flammable, so it will just go up in
Yes,
flames.
alcohol is flammable, and alcohol starts uh, uh, um, gets converted into gas very fast at around 57 to 65 degrees. So even little bit of heat also if we give, it will turn into gas, and that gas will catch up fire. So we do this double boiling method. This called as a double boiler. You know, you must have seen in your house your mama or someone making chocolate. For making chocolate also they use this method. So what they do, they don't heat the chocolate directly, they'll put it in some double boiler, some another container containing hot water, and then so that it does not get charred. So different uses of it. Here the use is because your alcohol is uh, flammable. Okay, so you're going to take a beaker containing hot water. In that hot water, you're going to put a transparent tube or something. If you have a tube, very good. If you don't have a tube, you can take a thin glass. You can take a thin glass containing alcohol, and in that alcohol, you're going to insert your leaf. The experiment that we are doing is because we want to find out the contents of the leaf. So that you're going to do, you're going to pick up fresh green leaves. Okay, and you're going to put it into the hot alcohol solution. You have to keep that as it is for some time,
Ma'am,
say for 5 minutes. Yes,
ma'am, but I don't have like iodine solution and beakers and all that. So how do I do it
so
then?
that's what I said. You don't have to see. This is how we do it in the lab. Okay, right now we are going, because of the current situation, we are doing it in a different way. So you can just take any container containing hot water. In
Mm.
that you put a transparent glass or something, a very thin glass, in which
Mm.
you can put your alcohol and you can put the leaf, and it can be just sitting there for 5 to 10 minutes, so that your alcohol also gets hot. And when do you know when to remove it? I'm going to tell you. Your alcohol is transparent in color. Okay.
So, ma'am,
Alcohol you don't
alcohol
have.
or sanitizer?
Alcohol you don't. Ah, yes, that's what I'm saying. The alcohol you don't have, you can use your sanitizer.
Ma'am, I
Okay.
have
You can
alcohol.
use
I just use alcohol. Is that fine?
alcohol. Not. I'm not talking about drinking alcohol or
No,
any
no,
of such
ma'am. I'm
alcohol. I'm
talking
talking
about
about the
I,
sanitizer alcohol.
yes,
Yes,
ma'am.
yes.
I
So
have
I
that.
need the.
Ma'am, we don't
Okay,
need
you have
iodine
that.
solution. We don't need
Ma'am,
iodine
We
I have
solution.
spirit.
Yes,
So,
I'll tell you the use of your iodine also solution. Wait, hold on. Okay. So you're going to now put your green thing into that. We need a transparent alcohol. Why do you need a transparent alcohol? So that the green color solution will your entire tube will become green in color. Okay, you will understand that the green color. So what was that green color that came out into the alcohol?
Chlorophyll.
Yes, so your chlorophyll came out. And I used what material to extract my chlorophyll?
Spirit. Ethyl alcohol.
Alcohol. Yes, your alcohol is important. You can't do this with water. Alcohol helps to break the cell wall. So the cell wall, that is a cell, each and every cell has got a cell wall. That cell wall breaks very easily with the help of your alcohol, and that is why we use a alcohol. Okay, so now there is there's a name. There's Nicola ma'am. You can pin yourself to Nicola ma'am. You can see there is Nicola ma'am. She will explain you the entire process. She'll show you the entire experiment. So I'll hand over to
Yeah,
Nicola ma'am
students
to
can
perform the experiment.
keep pin to KitKat itself. I'll be pinning Nicola here so they can view it.
Okay, okay. So you can keep yourself pinned to KitKat lab itself.
Ma'am, I have surgical this clinical spirit. That's fine.
Very good. Yes, yes, very good. You can use that.
Rubbing
Very nice.
alcohol. Yes. So I'll just use
Yes.
that.
Rubbing alcohol. Yes,
Yeah.
you can use that.
Okay. Uh, am I visible to everyone?
Yes.
Yeah. Is the
Yes,
setup
ma'am.
visible? Okay. So, in this experiment, we'll learn what are the components of the leaves. Okay. So, for that, you need a beaker, you need a tripod stand, you need a wire gauge. Okay. This is a candle. You need a test tube holder, and fresh green leaves. Okay. You also need a matchbox so that you can light the candle. Now, what you have to do, you have to take warm water. Okay. So, you have to heat the water with the help of the candle. Then what you will do, you will put some green leaves in the test tube. Okay. Now you're going to add alcohol to it. So I'm using ethyl alcohol. Y'all can use sanitizer. Okay. Okay. This is my test tube. I'm using a dropper and I'm adding alcohol. Okay. I've added the alcohol. Now what I'm supposed to do, I'll put it into this boiling water and I'll hold it for some time. Okay. You will keep it in the water till the green color, till you can see that the alcohol has become green. Okay, it will indicate that the chlorophyll has been extracted out.
Kids, while the green color is coming from the ma'am, y'all can start doing your process also. So y'all can also take hot water, and in that you can put a transparent some transparent glass, and in that you can put alcohol or uh, sanitizer, and in that you can add leaves. I'm sure
Ma'am, I have
you
this
must have
test
gathered
tube.
all the material.
I have
Oh,
this,
lovely, very nice.
so I
Okay, very good. Good,
boil water.
Atharva. You can put it in. It's hot water, no? It's nice hot and
Yes,
boiling water, right?
hot boiling water.
Good.
Just
Yes. So y'all all can do the same thing while ma'am is doing it, so that it will take less time and we'll be able to discuss the contents.
Ma'am, my
For today,
subject is
for today, if y'all don't have a transparent also container, it's fine. Y'all can use some other container also.
ma'am.
Okay.
I used a bowl and I put like a glass inside it, and I'm gonna put the
Okay, very good.
alcohol in here and the boiling water
Yes,
here. Will that work?
yes, yes, definitely. So we are just asking you to take a transparent thing so that you can see the green color coming off well. That is why.
Students,
ma'am,
please
this
keep
was
your
the alcohol.
videos on.
This surgic.
Good, nice, Atharva. You've got everything, test tubes,
We
everything.
bought
Good.
it. Yes, ma'am. We bought it before when they said alcohol based. You need alcohol based
Ma'am,
sanitizers.
Yes,
I don't have
yes, Harshini. What?
the I don't have a test tube or a beaker or any thin glass. I only have the leaf and
Ma'am, is this
sanitizer.
fine? Is this fine?
Yes, so
Boiling
you
water
can
and put and put
you can do
alcohol.
it in normal glass also. The glass that we drink at home, or you can do it in a normal another container also. It's not necessary that you want a transparent one.
Okay.
In any vessel you can take hot boiling water. In any vessel you can take hot boiling water, and in that you can put another vessel that will contain the alcohol and the leaf.
Okay, thank you.
Yes.
So I would like to share how I did this experiment. Okay, I was like all of you. I did not have, I did not have a beaker or a test tube, anything. So I took just two green leaves, okay, and I chopped them into pieces. And I took the chopped leaves in a katori, okay, in a vati. I added transparent sanitizer to it. And then I took one vessel and I poured water into it and I kept it for boiling. And I placed this katori with leaves into that boiling water. And after 5 minutes, I could see that my the sanitizer had changed color. It from transparent, it had become green. So you can do this.
Ma'am, my this test tube is plastic. Is that fine, or do you need glass only?
Better to have glass one, but next time don't use it to boil something. Okay, you're putting in hot water, it's fine. Don't put it
No,
in a
ma'am.
don't.
That, that would have melted the plastic. That's why I'm
Yes,
trying
yes.
it over here. Mine is greening. Well, it was
Okay,
orange.
can you see the color? I'll remove the leaves. Okay, so that you can see the color of the alcohol. Can you all see the color?
Yes,
Is
ma'am.
it
We can see the color.
what's the color?
Yes, ma'am.
What's the color?
Green.
Green.
Green. Okay. So it indicates that the chlorophyll has been extracted out of the leaves. Okay. Okay. So here we, apart from chlorophyll, there are also other pigments, but chlorophyll is a major pigment. Therefore, it is visible. So we extract chlorophyll in this experiment. Here. Have
Okay.
you not understood how to
So
do
now,
it? Yeah.
so we'll
Okay.
again go to our presentation. Okay, we'll go
Ma'am,
to our, yes.
ma'am, for me the leaf is not, ma'am, the color is not changing of the alcohol, ma'am.
Has your alcohol become nice and hot? Can you
Yes,
just
ma'am,
touch it with your
Ma'am, even mine
uh
isn't changing. It's
isn't changing?
no,
no.
ma'am.
Okay. You all can try it once more. Okay. You next time make small pieces of the leaves and then add it.
Okay.
Ma'am, I've
Okay.
done that
After,
only, ma'am.
acha, you have done
Ma'am,
that only.
yes, ma'am.
Yeah, ma'am. I cut the leaves and then I put a whole lot of sanitizer on it.
Um, can you turn your video on?
Look, ma'am. Here, boiling water and the leaves and the sanitizer, but the color hasn't changed.
See, the
No, has
extraction of chlorophyll, I mean, the color change will not be as dark as how ma'am had showed because she had used pure alcohol.
Hmm.
Okay. And
Okay.
at times it also depends on the leaf that you take, right, Farida?
yes. So you that's exactly what I was trying to say. That you need very nice and fresh leaves, very nice small soft leaves. So that the extraction becomes easier. You need to
Yeah.
pick up the new leaves, not the old ones.
Yeah, ma'am. I took it from my garden. The lily leaves from my
Okay.
garden.
Okay. Okay. So, can you touch the alcohol and tell me whether the alcohol is hot or no?
Yeah. The alcohol is hot.
The alcohol is hot. Okay.
Yeah.
Probably
Not
the
very
sanitizer
hot.
that okay.
Not very
It
hot, but
could be
still
probably
hot.
the sanitizer that you're using right now is not sufficient to remove it, and it's not even reached the temperature. Okay.
Hmm.
Probably that could be the case. Okay. So that is why we have shown you the process how it happens in the lab also. So, if it is not working with your place, you can see it how it happens in real life also in the lab.
Okay,
Uh,
ma'am.
the
Okay.
sanitizer which I used has 70% alcohol and it worked with that sanitizer.
I'm using 95% alcohol, but still it's not happening.
Probably it probably it's because of the either the temperature or the alcohol content. Okay. You can once this class is over, you can try it once more. Okay. And you all can send us pics if it happens or no. You all can let us know. Okay.
Okay, ma'am.
So we'll go to our observation. What is this weird structure? And my God, such a big structure it is, right? Any guesses? Any guesses? What
Structure
is
of
this?
chlorophyll or plants.
Yes, this is the structure of your chlorophyll, the pigment, the liquid that we're talking that we have extracted. Okay, so when people have studied it in the lab, they've come to know that this is how the structure of chlorophyll is. Can you see there's magnesium in between? Okay, so I told you all vitamins, minerals are very important your mineral, which mineral is being used in the chlorophyll? Magnesium. If there's no magnesium, then the green color will not be there. That is why if supposing you have got plants in your house, they tell you that if this mineral or if this kind of vitamin is not there, then there is a browning of the leaves, there is greening of the leaves, the borders become different, there are different diseases. Okay, it all depends on the vitamins and the minerals, that is the micronutrient that the plant requires. So one of that is magnesium. Magnesium is very important. Magnesium is present in your chlorophyll. This very big structure has got the presence of magnesium, which makes chlorophyll active. The magnesium is the one that is able to make the chlorophyll active, which picks up the sunlight. Okay, next.
Ma'am,
Yes.
ma'am, the sides of my leaf became brown in color.
Yeah, ma'am, my alcohol got like a really faint type of green, like you can barely see it, but yeah, it did get green.
It did get green. Okay.
Like,
Probably try to keep your water a little bit more hotter. Means
ma'am,
keep that same
is it
thing in more hot boiling water. Yes, yes,
okay?
yes. Okay. So your leaves got brown. Probably there was too much of boiling, so it became there was another chemical reaction that happened with it. Probably. Okay. Yes, I can see it's kind of got burnt, kind of burnt. Okay, you can try it once more, and then let us know on the group. Okay, so we'll go to our next slide. Okay, so if you remember, ma'am just said that besides chlorophyll there are other pigments also. Okay, so besides the green color pigment, there is yellow pigment, there is orange pigment, there is red pigment, and there are different names to it. The green pigment we call it as chlorophyll. Okay, in chlorophyll also there are many types of chlorophyll. There is chlorophyll A, chlorophyll B, and so on. There are different types of chlorophyll also. Okay, how and what they do function is different chlorophylls pick up different wavelengths of sunlight, different how do I explain wavelength would be the sun's energy that is coming to us is coming to us in forms of lot of waves. So different waves are being caught by different types of chlorophyll. So there is chlorophyll A, chlorophyll B is there, and so on. Okay. Besides that, sometimes you must have noticed that the leaves in different seasons change color. They get yellow in color, sometimes they get red in color, sometimes the leaf itself are red in color, and they don't appear green. Okay, but what happens in that case? The chlorophyll is present in the leaf. It is not that the chlorophyll is not present in the leaf. It is present in the leaf even if the leaf is red in color, yellow in color, or orange in color. It is just that at different times the plant decides which color it wants to show maximum. So the other colors gets masked away. The other colors get suppressed. Okay, so mainly in summer season there is nice hot sun, so what happens? The leaves are green in color. They do lot of process of photosynthesis. When it becomes wintry season, they may change yellowish color. Autumn, yellow, and then winter they become reddish in color. Those are because of certain types of other pigments that are also there in the plant. They are. It's not that they are not there in the plant. Okay. So
Ma'am,
the
maple leaves are like red or orange all year round.
Yes, they're red or orange all year round, but still they have chlorophyll in them. They
Hmm.
have chlorophyll inside them. Okay, and there are various techniques to detect that. There's one technique that we do is called as chromatography. In chromatography, we can see which pigments are there, and we can extract the pigments very well. Okay,
Ma'am,
so
so
the name, yes.
each type of plant has all of four of these, but the color is based on which one is the most main one.
Ac- yes, active. Yes. And same way, these names have been taken from where they are found. Like, for example, orange color is there in your carrots also. So that is why the name of that is carotenoids. Okay. Your red color is there in your red cabbage, in your red beetroot, okay, red flowers like your hibiscus flower. So those are called as anthocyanins, okay, and yellow ones are called as xanthophylls. So in each colored pigment, each colored part of the plant, these pigments will show their colors somehow or the other. Okay, but remember, even if your leaves are not green in color, they do contain chlorophyll, otherwise they would die, right? So chlorophyll is very, very important. Okay, next. So here, uh, after we do this, we have to write the observation. So what did you observe? What did you observe from ma'am's experiment? What did you observe? Tell me.
Leaves
What did
have
you observe?
leaves have chlorophyll, and you can extract them using hot water and alcohol.
No, using hot alcohol. Hot water that we are using to making to make the alcohol hot. By using
Oh,
hot alcohol, and what the alcohol does, the alcohol helps to break the cell wall to extract the chlorophyll. Okay,
uh,
so this observation, once you print out the sheets, you can write this observation down that you have observed today. Okay, so leaves contain chlorophyll. Chlorophyll helps us to prepare food in the presence of sunlight. They use water from the via the roots. They use carbon dioxide from the air, and this process is what we are calling it as photosynthesis. Okay, next. Okay, we've got another experiment also. Okay, in this second experiment, we'll see what is the aim. So next. So the aim of this experiment uh, is for y'all to, you have to keep it ready if you have a plant in your house, and then you're going to do this experiment, and then show us and click us the pictures and let us know what happens. Okay, here we are trying to show that plants cannot make food in absence of sunlight. Now, how can we do that? How can we make the plants not get the sunlight? Okay, so what you're going to do is you're going to take a leaf. On the leaf, you're going to tie a black color paper. Okay, and after tying the black color paper, you're going to leave it in the sunlight for some time. So now what will happen, where there is black color paper on the leaf, that leaf will not get that leaf will not get sun for some time. So you're going to leave it like that. You can keep it like that for 3 to 4 days. You can keep it like that. Okay, see, the first process in this is called as, we keep it away from sunlight for 72 hours for destarching. Means what happens is, once once you remove the plant from the sunlight, you keep it out of the sunlight, the plant, whatever reserve food it has got, it is going to use it. So that process is what we call at destarching, what is written the first process. Okay, but instead of doing that, what you do is you take the leaf, you tie a black color sheet, and you're going to leave it like that in the sun for 3 to 4 days. So in 3 to 4 days, what will happen is that the part that is black will not get the sun light. Okay, then what do? You do a you do a experiment that we call as, to test the carbohydrates. To test carbohydrates, we use iodine. So now you'll remove the paper. Once you'll remove the paper, you're going to put iodine on the leaf. Where you put the iodine, okay, so the black part where the strip was there, that part will not turn blue black. The other part will turn blue black, because iodine in presence, iodine is normally yellow in color. Ma'am will be showing it to you all. Okay, so iodine is yellow in color, but once you are putting it in presence of some carbohydrate, it is going to turn into blue black color. So the entire leaf will become blue black. Only the part of the leaf that you had covered, that did not get the sun at all, will remain as it is. It will not change color. Okay, so this experiment ma'am is going to demonstrate to you. You hold on, ma'am will be showing it to you.
Ma'am, ma'am,
Yes.
Ma'am, if we don't have iodine solution, can we use something?
Yes, yes, tell me. Normally you get iodine solution in the what do you call chemist. It is called as iodine tincture. It is used to treat some wounds. It's very easy to treat wounds. So normally iodine tincture is what is available. Try to find it out. It should be easily available.
Ma'am, and if we don't have that, can we use something else? Like instead of that?
No, you need iodine only. You can't use something else.
Okay, ma'am.
Yes.
Okay, so the aim of this experiment is that sunlight is essential for the plant to prepare its food. Okay, sunlight is very much essential for the plant to prepare its food, and without sunlight, the plant is unable to prepare food. So for that, we need a potted plant, and on one leaf, you will put black strips of paper on both the sides. Is it visible?
Yes.
Okay,
Yes, ma'am.
you have to fix it with the help of these paper clips. Okay, you'll fix it to one of the leaves and you leave it in the potted plant. I have only taken a leaf, but you all have to do it with the potted plant. You all do not have to remove the leaf of the plant. Okay, and you will keep it in the dark for 72 hours. That means you have to keep it in the dark for 3 days. Okay, after keeping it in the dark, what is going to happen? The food that the plant has already prepared, the starch that is present in the plant, will be used up by the plant. Then what will happen? After 3 days, you put it in the sunlight for 3 to 4 hours. After that, you have to remove these strips, these black strips. And you carry out the same experiment that we carried out just now. What was that? We used a tripod stand, a wire gauge. We put the beaker with boiling water. Okay, we have to use a test tube and a test tube holder, and you will remove these paper clips. Okay, you will remove the black colored paper clips, sorry, the black color paper strips, and you will put this into the test tube. Here you will put it into the test tube containing alcohol, and you will carry out the same type of experiment till the chlorophyll is extracted into the alcohol. After that, what you will do, you will remove the leaf into a dish containing water. You will wash it off properly. Okay, then you will take out this leaf and you will put it in another container, and you will put few drops of iodine in the entire leaf. What will you observe? You're going to observe that the entire leaf has turned into blue black color except the part which had the black strips, because the part which had the black strips, they could not prepare food because of the absence of sunlight. They could not get the sunlight, so they could not prepare food. And food is prepared in the form of glucose, and many glucose molecules combine together to form starch. Okay, so when the leaves could not prepare food, that portion of the leaf will remain unchanged. The color of that portion will remain unchanged. The other parts will turn blue black because the other parts of the leaf could prepare food. But that part, they could not absorb the sunlight, so they could not prepare food. So no starch is formed. Okay. See, the iodine solution, what is the color? And let me show you. Okay. It is a somewhat orange brown in color. Okay, this is the color of iodine solution. But when it comes in contact with the leaf, okay, where sunlight, which could absorb the sunlight, that portion is going to turn into blue black because starch is present there. And when iodine combines with starch, it forms a complex that is blue black in color. All right, did you all understand how to do it?
Yes, ma'am.
Are you clear?
Yes, ma'am.
Okay. So you all are going to carry out this experiment, okay? Do you all have a potted plant at home?
Yes, ma'am. I have a garden.
Okay, very good. So,
I have three
Yes,
pot
ma'am, I
plants.
have a potted plant.
all right. Okay, so y'all can carry out this experiment, right?
Okay,
Okay,
ma'am,
fine.
can you show us how the leaf turns black brown? You have got a normal leaf, no? Can you put the iodine solution and show us the result?
Yeah, but okay, I'll.
The leaf that you extracted from the previous experiment, you can use that only.
Okay, so these are the leaves that chlorophyll was extracted out. Okay, so we put iodine solution.
Yes, we can observe that the leaves are turning blackish. That shows that the leaves contain starch inside them. Okay, so this is what you all are going to do. See, kids, related to plants, the experiments take time because they are plants. We cannot do the experiments very fast. So all the experiments that we'll be doing with the plants related would be that we would tell you to do it and you will have to do it by the next week. So that the next week you all can show us the results, because you can't ask the plants to do it immediately. Okay, chemical reactions, physical experiments, you can do it immediately, you'll get the results very fast. But bio related topics will take a little bit more time.
Ma'am,
Okay,
my thing turned green. The alcohol. See.
the first one turned green. Very
Yeah,
good.
the
Did
alcohol
you
turned
do
green.
Okay, do you have iodine solution ready? You can put iodine into it, so you'll be able to see if your leaf is turning blue, black, or no.
Hmm. Okay.
I'll go down and get iodine.
So the observation in this experiment is that you will be observing it that when you put iodine on the part that was not covered, it becomes blue black, but the part that was covered will not turn blue black. Okay, so very important, because the part that did not have that had the black strip, that was away from the sun, and that part did not carry out photosynthesis, and so that part did not get starch. Okay, so for photosynthesis, sunlight is important, and this experiment we carry out to show that sunlight, yes, it is important. Can we go to the previous slide? Yeah. So this is exactly what I was telling you, chlorophyll A and chlorophyll B. Chlorophyll A will absorb sunlight at these numbers. You know, it is called 413 nanometers, 616 nanometers. So they are different ranges of the wave that we get from the sun. And the chlorophyll B is taking it at 450 and 640. So two different types of chlorophylls are present in the plant, and they are going to they are going to absorb the sun's light at different wavelength. That is very important. Okay, so this you can see in the leaf is very thin, but it has got many layers. There many layers are there, and in that layers, if one particular cell that you see, one particular cell will contain the chloroplast, like you can say like a building chloroplast, which will have the pigment chlorophyll inside it. Okay. Okay, so we'll go to a question session. I first I want to ask that whether y'all have any questions related to this, any topic. Y'all have got anything, any questions related
No,
to this?
none.
No, nothing.
No,
No.
ma'am. No,
No. So
ma'am.
did you enjoy this? Did you enjoy this
Yes, ma'am. I love it.
session today? Did you feel it
Yes,
informative
ma'am. I
today?
enjoyed it.
Yes, ma'am, we enjoyed a lot.
Yes,
Was
ma'am. It was
was it
really
new
nice.
information, or did you already know it? Yes, you got more
Ma'am,
information,
No,
already.
no,
right?
I
So
got
science
lot
is
of
always like
new
that. You
like.
Ma'am.
Yes. So what is science? You know,
Good.
every day you have to learn something new. So today I told you something new. You can go and research about chlorophyll A, chlorophyll B. You can research about all the pigments that we just saw pictures about. You all can see the structure of chlorophyll. Along with that, I'll tell you, go and see the structure of hemoglobin also. Hemoglobin, you all are going to tell me which mineral is there in the hemoglobin, the next class. Okay, you all are going to tell me that.
Can I
Okay?
tell? I already
Okay,
know.
ma'am.
No, 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 no,
Yeah.
no, 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 no. Please keep it for next time. Okay? If you all know, keep it with yourself. If you don't know, please research it and tell me next week. Okay? So we'll go to the questionnaire session. I will ask you a few questions. You all are ready?
Yes, ma'am.
Yes,
Okay.
ma'am.
Okay, so there you go with your first question. Your first question says uh, there are some leaves that has got white and green patches all over them. What is the name that you give to those leaves? Do you call them strippy leaves? You call them diseased leaves? You call them patterned leaf, or you call them variegated leaf?
The D, variegated leaves.
Variegated leaf. Very good. Okay, good one. Variegated leaf. Okay. Now you all are going to tell me which component is not required for photosynthesis.
Ma'am, oxygen.
Yes, oxygen is not required for photosynthesis. You did not give me time to give the options. Okay, very good. Okay. Um, which metal is there in which metal, or I can say which mineral is majorly important for the activation of chlorophyll pigment?
Magnesium.
Very good. Okay. And one last question for you all. Okay. Where is chlorophyll present in the cell? Where is chlorophyll
Chloroplast.
present in the cell?
Chloroplast.
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 Yes. Very good. Okay. Smart kids. Very smart kids. Good one. Okay. Okay. So today's session, even I enjoyed with y'all. Okay. And we hope to meet you next time So next time a little bit preparation you all will be requiring to do. You all have to keep a moist bread so that fungus grows on it. And then we're going to study the structure and the fungus uh, m- a little bit about it. Okay. So we'll be putting the process on the group. You all can follow it. So by next week you all are ready with that material. Okay.
Okay. Okay, ma'am.
Okay. So Swapna, we're done for today.
Okay.
Yes, yes. All right. So, students, we will continue. So, the second activity that we did today, you all will perform it at your place and you will record the observations. Okay. And as ma'am said, for the next week, you have to keep a bread uh, to grow fungus on it. We will share the process. So, these two things you should keep it ready for the next class.
Okay, ma'am.
Okay. All
Bye.
right. Bye bye. Thank you so much.
Okay,
Bye,
ma'am.
ma'am.
Bye,
Thank you.
class.
Bye.
Thank
Bye.
you.